So Austin is, if you remember, an international partner at the Argonne National Lab, and we've been collaborating with them over 2 or 3 years. But now we suddenly met again in person at the ISC meeting in Hamburg, didn't we, Austin? And you are going to tell us about some of the things that have been going on in that kind of collaboration from your end in terms of the COVID-19 drug discovery activity.
All right. Well, so as I said, good afternoon. Thank you for the introduction, and thanks for having me virtually. My name's Austin. I'm an assistant computational scientist at Argonne National Lab in the Data Science and Learning Division. And today I'm going to be talking about kind of a couple things. So the first thing I want to talk about is kind of summarizing some of the results that we've had from computational experiments from COVID-19. I actually spoke at a couple Comp Biomed conferences in 2019 and 2020 on what we were doing. So now we have kind of the closeout results of that. I want to talk a little bit about some late stage drug discovery things that we could do. I think we focus a lot on kind of AI and infrastructure for early stage for drug screening, but we don't talk too much about once you find a lead, what can we do with it? And then lastly, I'm going to kind of close out on talking about what are some of the infrastructure requirements that we might want to think about in the long term, given we want to expand on computational drug discovery programs. So this work was done kind of in collaborations with international partners and mainly through collaboration between all the United States national labs and what was bundled together and called the National Virtual Biotechnology Lab. There was lots and lots of people involved in this. And it actually had a couple different components to it, not just molecular design, which is the one I'm talking about. This had to do with like epi models and different types of R&D setups. And what was very fun about this collaboration is we worked very, very closely with kind of all the different groups. So everything from neutron sources and light sources all the way to high performance computing. And we even had laboratory assay staff involved in this. So part of this screening effort was trying to computationally identify initial lead compounds from large databases against COVID. And at the time we were doing this in 2020, we didn't have structures early on for many of the different targets. We didn't actually know what targets would be beneficial to target. Initially, we started out looking kind of at the entire set of 13, 14, 15, 16 proteins. And so what we ultimately did is we were able to do some modeling on these, get some structures, and begin the design process. So the workflow that we used was standard computational docking for drug discovery, protein ligand docking. And in order to speed this up, to try to screen the large scale libraries that were available at the time, which were, the in-stock libraries for compounds you can order kind of all over the world is around, I think it's like 5 to 12 million compounds were the ones that we know we could get shipped to us in 2020. And so the overall structure of our experiments were pretty straightforward. What we were doing is we were taking structures. So we were mainly looking at the main protease. We were taking libraries that we knew we could order from, and we were basically docking. And we were using AI to do accelerated docking. So one thing that we had is we had multiple structures from the main protease that were resolved at different sources, different resolution. We had from the diamond light source a bunch of fragment libraries. And so we wanted to use all of these in a consensus docking setup. So this required some extreme computing, which we used various resources in the United States, from the Argonne Leadership Computing Facility with Theta to a couple systems actually at the Texas Computing Center. Initially, we started off by just using some automated binding site detection protocols, which at first we were quite skeptical of, but it turned out, when you're trying to do these large scale compute campaigns, they actually work decently well to identify sets of pockets to explore. I'm going a little quick through this, I should say, just to summarize kind of the results and get to some more interesting components of the talk. So when you do a large scale computational docking screen, one of the challenges is what you get out is not, these are the drugs you should purchase, assay, and test. What you get is distributions over millions of compounds. And so one big challenge, and I think is still an open question, is how do you actually end up taking this data and informing the choices that you pick to actually assay and buy? So this was what the data basically looked like after we did all the computation. So we had multiple different structures for the main protease that we docked all the compounds against, and then you have these kind of large distributions. And so the question is, how do you pick compounds? So we actually did something, probably in hindsight, I think it's a little naive, but what we did is we essentially just took off the top 1%, did some diversity sampling. You essentially cluster all the compounds, pick representative clusters, and order those. One difficult thing with this consensus docking, as you saw on the previous slide, that the distributions don't exactly overlap, is even across different crystal structures of the main protease, docking into the same binding site, you actually don't get a lot of correlation. So what you're looking at here is just a scatter plot of a couple compounds that we sampled the data set of. And we're just showing how even across different structures, you end up with some correlation, but not a lot. Especially when you're thinking about how do I pick, top scoring compounds, which are, I don't know if you can see my mouse, but are these compounds way to the left bottom corner, the more negative is a better score. It ends up being quite difficult. But ultimately, we ended up testing about 100 compounds, and about 50 of them had some activity. We did identify one compound which had an IC50 of 4.2 micromolar. And we were able to get crystal structures, and we were quite pleased to find that the predicted structure ended up mapping on quite well to the experimental one that we derived. So as a part of this, we had many different groups working on it, and there was also, I should talk first about the computation. We ended up computing over 300 million docking scores. And part of this is trying to figure out now how to release this data in some useful way. As I'll talk about later on, one of the challenges is building AI models to accelerate docking, especially when, as you saw, minor changes in numbers. So, a little bit of an error greatly affects how you sample that to pick what compounds you buy. If you slightly miss a compound that's in like the top 1% of your docking run, then you're going to ultimately not buy it. And so the statistics for how we think about AI models is not thinking about the normal use case where you're looking at like a normal distribution and you want to model the behavior around the kind of mode of the distribution. What you're really interested in is modeling the extreme of the distribution, because you actually don't care about specificity around the mode when you're thinking about docking, because if something has an average score, you're not quite interested in it. So this is kind of ongoing work in which we're thinking about. This was also coupled with high fidelity simulations. This was also in conjunction with some of Peter's group and Arvind Ramanathan at Argonne. So we were able to do simulations on these compounds, on some of the top scoring ones, identify novel states of these compounds. This is using Deep Drive MD, which Arvind developed. And as you can see, what Deep Drive MD does is essentially take a look at states during simulations and restart simulations using an AI-driven model to try to identify behavior. So I want to talk a little bit more about what I think is a slightly more interesting question in this area of computational drug discovery is we found a lead, but now what? What do you do? Do we all of a sudden give up on the AI methods? So there was some sort of, I like to call it professional DUNSAR, which is just done by medicinal chemists. I say this as a computational scientist on this compound, and they were actually able to get it to get the KD value down substantially by swapping the dichloro part for this triple chlorine. So it was exciting to not only find the compound, but find a compound which had decent SAR. But it got us thinking a little bit about, well, how are medicinal chemists designing these kind of, series, right? You look at something like this, and you can kind of naively say, well, you maintain the scaffold, you sort of change around a couple functional groups to optimize the structure. Once you get something like a Newton structure, you can see where there's space, you can see where you might want to alter things. So it got us thinking about the way that we do docking. So the way that computational docking is traditionally done is you have a list of compounds, you treat it like a bag of things, you pull something out of the bag, you compute the docking score, and then you put it back in the bag with the score attached. But we know that from like fragment-based drug design, and we know from SAR that there is some internal structure to the way that these molecular databases are organized. So what we're depicting here, for example, is we have a compound here, which is a compound you maybe pull out of this database, right? To think about this bag, and you dock it. But really, you can also think about its constituent components. And if you have the constituent components in the right spot, so the same spot that it would be in this big compound, you can imagine a sort of tree structure where you're slowly working up from, we have these fragments, we combine them together, we combine those together, you combine that together, and eventually into a scaffold, and eventually you get the compound. And so it got us thinking of, well, this is a tree structure now. We know how to do things we know how to do machine learning on different types of structures like graphs. We normally think about molecules individually as graphs, but you can also think about the decomposition of a molecule scaffold as a graph, a kind of hierarchical global graph. So we came up with this kind of mental model of thinking about scaffolds as being kind of big bubbles that contain inside of them compounds. So a famous example is the penicillin class of compounds. So amoxicillin and penicillin actually share the same scaffold. They just have slightly different functional groups on them. And different types of antibiotics, such as penemnocycline, for example, shares the scaffold actually of penicillin. It just has a couple different modifications to it. And so thinking about this kind of graph operation, we formalize this into a lattice structure. So for those familiar, a lattice is just a kind of ordered set of relations between things. So we could basically think about navigating chemical space through scaffolds. So we came up with some operations. So if I'm sitting on a scaffold here, I could think about its predecessors that are kind of smaller components of it, or I can also think about successors, which are larger scaffolds. And so you can imagine taking a chemical database and making it into a giant tree like this. So each of these are a molecule, and it's linked up to its predecessors and to its successors. And this is just illustrating some score of how you can kind of go from smaller components, which small fragments don't often have great binding scores to proteins, all the way up to kind of larger compounds. And what we did is we actually trained a transformer model to walk around the structure. So essentially, the way you can imagine it is if you're sitting on a node on a graph, you're sitting on, say, that penicillin scaffold, you can actually tell this transformer to say, hey, can you generate the nodes above you? Can you generate the nodes below you? So why would you want to do this? Well, for those who've worked on HPC before, you can imagine if we tried to store our entire database of millions, possibly billions of compounds in a big global graph structure that we would distribute out to nodes, that would be a massive communication problem. And the interconnects are really not able to keep up, especially when you're doing something like ML inference, which is very, very fast. So instead, we thought, well, why don't we just train a transformer to be able to navigate that graph? So that way you can distribute this model out to your nodes, so you don't have to do this type of communication. And so this actually trained quite well. We were able to generate scaffolds very, very efficiently and accurately. And what's exciting about this is that it's a generative model that's guided. So it's not like the usual kinds of generative drug models that kind of just spit out like a bag of things that maybe have some association. This one spits out things that have a very strict association. And we actually took a look too at some of the assay data from the Diamond group and John Cadera's work. And we noticed that if you take some of these assays and you color this scaffold graph, you find that the performance that you get from these compounds actually maps quite well onto different clusters of the graph. So what we were building up is, you can imagine a big global graph structure of all compounds that's kind of walkable. So you can kind of do different types of graph sampling in random walks. And we are going in with a hypothesis that you can actually cluster this graph into scaffolds that are active and not active. So we call this scaffold-induced graph sampling. So you essentially are able to do docking on a big, large database without having to dock the whole thing. by having random walkers kind of walk around this big global graph structure and identify scaffolds that will most likely show activity. In a sense, this is like combining the previous steps of how do we pick compounds. So instead of docking the whole thing, doing all the computation, instead we say, well, why don't we just combine the two steps of when we try to do the diversity clustering. So you, at the end of docking, you end up with this big database, so you cluster it, you sample the hits that you want. Instead, we said, well, why don't we just combine them together?
Austin,
you
have 5 more minutes now.
Okay. And so we're also working with a visualization student on building this into a visualization interface. So you can imagine connecting this up to a high performance computer while this random walk is going on. You can actually have a med chemist, if you ask nicely, sit there and watch as this is happening to kind of steer the simulations too. So this is adding in some explainability to the way that these simulations and campaigns are run, and also giving a nice visual interface. We're actually working on putting this into VR, which will be hopefully fun in the future. And so then briefly, I just wanted to talk about kind of some of the lessons. So I guess this is kind of what we're working on and how we're thinking about improving some of these drug design campaigns. And last, I just wanted to talk briefly about the infrastructure component of, when we think about building supercomputers and what kind of hardware should we be building next for these kind of drug discovery runs, what should we be thinking about? And I think that, in the back of our heads also is, pandemics are seemingly happening more often, or at least it's on our minds more. And so I think there's definitely some funding going around for building kind of biopreparedness centers. So these might be surveillance programs for computational drug discovery where, you connected to epidemiological models and you're kind of, instead of waiting until it's a real pandemic and then starting the drug discovery, you're actually having an arsenal of folders where you're already prepared for different threats. So the way that we thought about this workflow analysis was essentially what you do is you take a database of a lot of compounds, you want to build a model that will be able to predict some amount of hits computationally. You can only assay, some of these. So these are our kind of two parameters of what do we call hits and what do we call the things that we go to assay and how do we get those to leads. And so as I said earlier, we have to pick cutoffs. At least that's the best method that we currently have for taking computational results to assays. And so the question is, well, how do we improve this? So I showed these plots before. So one thing that we often think about is for our AI models that take in molecules and try to predict the efficacy, we're not trying to predict the mode behavior. So we have to think about different ways of analyzing these models, right? A model that's 99.9% accurate at predicting the behavior around the mean of a docking distribution is just not useful. It might be, but that statistic is not telling of the question of, does this identify things that ultimately will be leads? So we've been using this type of regression analysis. It's really like an enrichment. So essentially what we do is we think about, well, what's our cutoff for our computational screen? And where are the hits that we want to identify living? So you can imagine, the top percentage of your database might contain hits if we really did all the docking, and we want to identify those. So ultimately, this, as I said, you can take 1 million molecules. You want to take 100,000 of them through computation, and you have to pick ultimately 1,000 of them. And so what we actually realized is that this relates very well to the accuracy of your models. So what we were able to essentially show is that if we think about the throughput of our surrogates models, so this is our GPU speed. So how much are we able to dock in a day on a GPU with an AI model? And we think about how accurate do our models have to be in order to get speed ups over the traditional workflows to screen more compounds. We actually realized that it's not the speed of our GPUs in our compute that's actually slowing us down. So what you can see here is this red line is showing you kind of the throughput that we can get on a V100. And what these red lines are showing you is the accuracy for our AI models. So this one red line here is saying, well, if this AI model can detect 90% of the top hits, we can get essentially a maybe 10x speed up over docking. If our models can detect better, then we can get a greater speed up, and we can get another order of magnitude speed up. And so what's fascinating for us as we think about designing the future computers is that actually it's not speed that's really the problem for us. It's designing new AI models. And so that's why we're happy to be releasing hopefully soon this large database of docking scores along with the assays to hopefully build new AI models. But it's going to be requiring a different way of thinking about how we train models for doing anomaly detection, basically, because we're concerned about extreme behavior. So I guess the takeaways today are that out-of-box ML models do speed up things and allowed us to find hits, but ultimately we need to think about new ways of modeling these. And we also need to be thinking about SAR and how we could be using that for AI. So thank you. Thanks for the lightning talk, I guess, on this. And I'm happy to have any questions and talk further.